questions, but I hope that you'll use the session after this for an opportunity to further interact. Thank you very much. It's now my great pleasure to welcome Phil Sharp to the podium to give our next talk on RNA as a gene regulation modality. Phil probably needs almost no introduction for this audience, except to say that really your work has provided the foundation for the RNA revolution, and it's one of the most important parts of my career. Thank you very much, Phil.
Thank you. It's an honor to be invited here to celebrate Govind Khurana and his colleagues. I also an honor to recognize Govind and Esther's friendship over the 30 years that we have shared being colleagues at MIT. And clearly, as others have expressed, we are all indebted to the science of Govind Khurana for much of what we understand now in biological science. What I want to do today is to talk about some new science related to RNAi. And if I have the pointer, there it is. As you well know, part of the revolution in understanding that RNA is a major regulatory molecule in mammalian cells and other organisms comes from the discovery of the widespread utilization of RNAi and microRNAs in the regulation of expression of genes in cells. And if you look at the pathway of synthesis of microRNA, Pol-2 is typically engaged in transcription. You make a hairpin, which is processed by Grasha, and then you get transport to the cytoplasm, where Dicer interacts with that hairpin to make an siRNA type intermediate. Argonaute loads that double-strand RNA and takes one strand and uses that to target messenger RNA, either in the context of translational repression or if there's exact complementarity cleavage. And siRNA, when transfected into cells, enter this pathway at this point, engaging Argonaute in either translational control or exact cleavage. And we'll return to Argonaute as an intermediate or component of the complex in the cytoplasm of cells. And what we're going to talk about today mostly is the issue of this translational repression process in the cytoplasm of cells. Now we know from the early work of Ambrose and Rufkin that the specificity for targeting microRNA regulation is this seed region between positions 2 and 8 from the 5' into the microRNA. And it's pairing through this seed region, and David Bartel gave us the term seed region, that determines the specificity of microRNA regulation for messenger RNA. Now, it's clear there can be some single nucleotide bulges in this sequence, but by and large, it's a seed sequence that is critical. And if you look for evidence of what fraction of RNA is engaged in regulation by microRNA in mammalian cells, the best estimate comes from the work of bioinformatics from Chris Burge and David Bartel, and I'll illustrate that here. What they did was to look through evolutionary comparisons of the 3' UTR and ask what is the frequency of sequences in conserved sequences over evolutionary time that is complementary to the seed region of the microRNA. And what they found was that those types of sequences, those evolutionarily conserved seed sequences, are present at about 3 times to 4 times more frequent than you would expect from random comparisons of any normalization technique. And that has led us to the idea and hypothesis, and I think it's very strongly supported, that about 55% of all the genes in mammalian cells interact with the 87 evolutionarily conserved seed families of microRNAs expressed in those cells. These are families evolutionarily conserved, expressed at reasonable levels, high levels in some cases, in specific cell types. About half of the genes show no conserved sites within the 3' UTR, and we anticipate that they will not be regulated extensively by microRNA, though there is emerging data from a variety of labs that microRNA can target sequences with encoding frames of messenger RNAs. But let's look at this 55%. And that 55% of genes have on average in the 3' UTR 4 target sites that are evolutionarily conserved for microRNAs. And these target sites typically are targeted by more than one family. So it's a combinatorial type regulation. And per microRNA in this evolutionarily conserved family, it appears to target about 50 different messenger RNAs. So what you see out of this is about half the messages in higher cells show evidence of evolutionary conservation through a network of microRNA interacting with those target messenger RNAs. And one of the great paradoxes is that this whole regulatory system, extensively evolutionarily conserved, appears to regulate messenger RNAs in somatic cells at 20 and 30% changes, maybe twofold changes is sort of an unusual change, at the level of translational regulation. So why all this conservation and yet such modest changes in the regulation in messenger RNAs? So I want to spend the rest of my time talking about one possible explanation for that paradox. And part of that, this explanation, I think comes out of the relationship between microRNA and stress. It's been published over a number of years that microRNA have a specific relationship with stress. Back in 2003, the microRNA 14 in Drosophilus seems to control responses to salt stress. In the very important work by Eric Olson, where he showed that microRNA 208, which is encoded within an intron, a myosin heavy chain, regulates the stress response to cardiac stress in mice. Under normal conditions, the mice show no defect. Stress them, then they show a change in response to stress. Same is true of microRNA 8 in osmotic stress in zebrafish. And this I like particularly well because it was done by a graduate student, ex-graduate student in the lab. microRNA 17 in eye development in Drosophila. There's extensive regulation and interaction between microRNA and a number of genes that control signaling and development in Drosophila. If you keep the flies at a constant temperature, there is no phenotype when you delete microRNA 7. But if you allow oscillations between 24 and 31 degrees, eye development falls apart. So it's clearly that this microRNA is engaged in response to variation in the environment and to the robustness of the system. Now, we published several years ago in 2006 that Argonaute, this key protein in the regulation of messenger RNA translation, can be found in stress granules dependent upon the presence of microRNAs in cells. And you'll recall that stress granules form after mammalian cells are stressed in most cell types. And there is translocation of proteins from the nucleus of the cell to the stress granule that's engaged in controlling the translation of the RNA and the response to stress and the half-life of messengers. Argonaute is also found in P elements, P granules, in the cytoplasm of cells. And I want you to recall that if you photobleach Argonaute in a stress granule, it exchanges within 20 seconds. If you photobleach Argonaute in P granules, it exchanges over 2 or 3 hours. So it's a much more stable component of cells and is probably committed to RNA degradation. This is committed to RNA translational regulation. And in the following way, that messenger RNAs, when they're in stress, some subset of messenger RNAs determined by RNA binding proteins arrest translation at the level of 40S binding, no 60S binding, and they tend to coalesce in stress granules in a dynamic way. And as they move out of stress granules into the cytoplasm, translation continues. So this is a very dynamic control of stress responses. And I want you to recall again that the proteins that are found concentrated in this stress granule is TIA1, G3BP. This is a RAS interacting protein that also binds to RNA. Poly A binding protein and Argonaute 2. And a number of other RNAs are found concentrated in this stress granule structure. Let me give you an example of what stress granule formation is like. You see here a cell, a HeLa cell in this case. It's got GFP, Argonaute 2 being expressed. Here's the Argonaute 2 in P granules within the cytoplasm of the cell. This is at time 0. You then stress the cell. Let me see if this will run. And then over 30 minutes, you see the formation of these other sub-cytoplasmic organelles that are stress granules. And what is present in those stress granules is Argonaute 2 and about 5% of all the Argonaute in the cell. So P granules here and then stress granules form over a period of about 30 minutes, in this case with arsenic stress, but you can stress cells by any one of these methods and stress granules will occur. Stress granules is also found in disease states, as we've heard. Here is the hypoxic region of a tumor, and that tissue has been stained for, in this case, it's GFP being expressed in those cells, and you see stress granules throughout that tumor region. This is the DNA and the overlay, and so stress is a common occurrence in cells, and stress granule proteins interacting with messenger RNA controlling translation is a common phenomenon. So what I'm going to tell you about stress granules today is that this polymer, ADP-ribose, is related to stress response in the cell, is probably required and controls stress granule formation, and is engaged in translational regulation. And we know that this polymer is involved in stress response in the nucleus through the extensive studies that show that poly-ADP-ribose, or PARP, is synthesized in the nucleus of a cell following DNA damage, activation of PAR1 mostly, use of NAD to modify glutamate residues on the surface of proteins. You get an extensive polymer that can be branched, and that opens the chromatin, probably targets repair activities to this region and facilitates DNA repair. And this process has been well studied in the nucleus of a cell. We know that PARS, poly-ADP-ribose, is essential in multicellular organisms. It's required for chromosome integrity, as I just mentioned. It's involved in transcription in ways that are not well understood. It influences cell division. And PARP1, this DNA repair activity, is highly targeted in the pharmaceutical industry because in BRCA1 cancers, BRCA1 being involved in DNA recombination, in those negative cancers, if you inhibit PARP, you get a remarkably good response. So PARP is PARP1, nuclear PARP, is a very promising pharmacological agent for treatment of BRCA1. What we've noticed over the past several years in other labs is ischemia, inflammation, and degenerative diseases and vascular diseases also are showing responses to PARP inhibitors, and we don't understand, and perhaps this will be part of the explanation. And I just want to point out the sort of size of polymer we're talking about. This is a two-dimensional electron micrograph on a protein film apart. It's just like RNA. You can visualize it in an electron microscope. This is a polyribosome, and these two things are comparable size. If this is being polymerized off of proteins that bind to RNA, bound to messenger RNA, it's quite an extensive modification. And this is the structure, as I pointed out before, pyrophosphate type bonds, but otherwise it's a very similar ribose rings, 2' hydroxys, adenosine, very similar to RNA. Now what I'm going to talk about is other parts than PARP1. This is PARP1 and PARP2 are involved in DNA repair. There's 17 parts, all defined by having this PAR domain that we think could be involved in poly-ADP polymerization. By and large, the activities of most of these PARs are unknown. And this work that I'm going to talk about in the next few minutes comes from the collaboration between Paul Chang, who just joined the Koch Institute at MIT, who's an expert and has developed reagents around all these PARs, and my lab looking at microRNA control. The first thing we noticed is that these stress granules have the poly-ADP-ribose concentrated in them. So here's an example in which cells in culture, HeLa cells in this case, under controls, being stained by antibody to the polymer, antibody to EIF3, which is involved in stress granules, and the merge. And you see under oxidative stress, the PAR is concentrated in the same regions as the stress granules and the overlay. And the same thing is true of other forms, other ways of inducing stress granules. Not only is the polymer localized and concentrated in the stress granules, but the polymerases are also concentrated in the stress granules. These are GFP-tagged polymerases. I can do it with antibody to endogenous protein as well. And what you see here is PARP 13 and 13.1 splicing isoforms of each other under stress conditions. This is staining with the antibody. This is staining with the EIF3 in the merge. And the same thing is true of PARP 5A, PARP 12, and PARP 15. 4 PARPs are localized in stress granules under stress conditions. The glycohydrolase, the polymer that hydrolyzes PARPs, is localized under stress conditions in the stress granules. So what this suggests is that there's a very dynamic hydrolysis and synthesis of this PAR in the stress granules during synthesis. So let's look at a few specific proteins. So this is Argonaute 2. As we know, it's targeted to the stress granules by microRNA. And this is a Western blot by PAR after immunoprecipitation of Argonaute 2 under non-stress and stress conditions. Argonaute 2 and PAR modified Argonaute 2, longer exposure. It's modified under stress conditions by poly-ADP-ribose, but it's not most extensively modified. Here is 4 proteins, Argonaute 2, which I showed you before, G3BP, which is a protein that localizes to stress granules, hydrolyzes, binds to RNA, and will also engage RAS in a very specific way. So this is a connector between stress, RAS activity, and translational control. And this is poly-A binding protein 1, and this is TIA1, which I showed you relocates from the cytoplasm to from the nucleus to the cytoplasm under stress conditions. If you overexpress Argonaute 2, G3BP, or TIA1 in the cells, you will nucleate stress granules without having stress. Poly-A binding protein, that's not the case. It localizes in stress granules, but it doesn't nucleate. And here's non-stress and stress cells being immunoprecipitated with TI1 and PAR, Western blot, and you see a dramatic increase in the polymer modification of that protein. You see overwhelming modification of G3BP binding protein that increases under stress. Argonaute increases twofold or threefold under stress, but poly-A binding protein does not show modification. So all 3 of these proteins that can nucleate stress granules are modified by poly-ADP-ribose in the cytoplasm of cells. So that leads us to the idea that stress granules are being formed by the formation of this poly-ADP-ribose, and that under stress conditions, PARs, which are bound to proteins that are engaged in stress response, are modifying Argonaute 2, modifying G3B binding protein, TIA1. There are proteins that bind specifically to PAR, and this forms a network in the cytoplasm of the cell. Well, the hydrolase hydrolyzes this network, and that creates the dynamic activities of stress granules. And this then becomes coalesced in a region. That's what we visualize when we stain cells. And if we overexpress the hydrolase, we don't form PAR stress granules. It will block the formation, hydrolysis of this. If you overexpress PAR, you will stimulate the level of stress granules. So we're pretty confident that the ADP ribosylation is involved in this process. Now, what does that mean for regulation by microRNAs? And here's an experiment that probes that question. What we've done here is to take luciferase with a very short half-life. This is a 30-minute half-life protein, so we can get responses if you're looking at stresses over the tune of hours. We transfect it into cells with target sequences in its 3' UTR that are targeted by a siRNA delivered microRNA. And we ask, what's the degree of expression of this luciferase in the absence of the microRNA and in the presence? And the only thing we're looking at here in this scoring is microRNA-mediated regulation, because we're normalizing under the same stress conditions or normal conditions to no microRNA. So what you see, let's look here, that under conditions of normal conditions, this system shows about 20-fold cytoplasmic repression, and that's because we got 6 of these targets, and we get a good reporter response with that. But if we use an siRNA to reduce the level of the hydrolase, and I won't show you that the siRNA is effective, we see a 7-fold reduction in the level of suppression by this microRNA. If we move over here, where we're using stress conditions, the first thing you notice is that the microRNA regulation is reduced. So there's stress on the cell is relieving specific microRNA translational suppression. And if we inhibit the hydrolase, increasing the ADP-ribosylation levels, we see an increase, even a further decrease in this microRNA suppression. So what we believe this shows, in a fairly direct way, is that microRNA translation regulation in the cytoplasm of the cell is regulated by this process of PARP synthesis. So under non-stress conditions, Argonaute 2 targeted by microRNA to messages inhibits translation, and under stress conditions, we're seeing in these cells, and it's PARP 13 and 13.1 that's specifically interacting with Argonaute 2, we believe by co-immunoprecipitation, that this is in some way relieving stress translation. Now, perhaps it's because this is cycling through stress granules and coming out, but there is an effect here on stress granules. So in conclusion, what I think our data says for the first time is that 5 PARPs and 2 hydrolase isoforms are localized in RNA-binding protein-enriched stress granules in cells. So this first relationship between poly-ADP-ribose and cytoplasmic stress responses. That poly-ADP-ribose is required for the structural integrity of stress granules, a dynamic relationship between hydrolysis and synthesis. That specific RNA-binding proteins are modified by PARP under stress, affecting gene, post-transcriptional gene regulation, particularly microRNA regulation is reduced, and poly-ADP-ribose probably regulates translation under normal and stress conditions, suggesting that this polymer has a much more extensive role in the biology and cytoplasm of the cell than we had ever anticipated. It's engaged in nuclear processes, but I think it's engaged in very dynamic processes in the cytoplasm of cells. And this work was done primarily by Anthony Lung, a postdoc in the laboratory, and it was done in collaboration with Paul Chang, whose work on PAR and PARPs made all of this possible with CJ, his graduate student, and I thank them.